Okay. So are there people from group one in the room right now, who could present what they found when doing their part of the code? Mm. Or are there people from group one who have questions about what they were doing too? That's all. That was the other alternative, is just ask questions about what you were doing. Oh, great. Thanks, Tina.
Yeah, hello. Good morning. So, please, can you hear me?
This looks great, Tina. We can hear you too.
Okay. So, for group one, we were looking at the PCA analysis, interpreting the PCA analysis and some of the biological information that we deduce from the various principal components. So, so So for what we did with the codes that we ran, we found out, so this is like the elbow plot of the various PC components and its standard deviation. So as you can see here, so when we look at the graph, we can see that at PC component 10, 75% of the data is represented as PC component 10. And as we move forward, we can see that the variation between as we move from, let's say from 12 going, we can see that the variation is decreasing. So we decided to go with PC components 1 and 2 since they have like the highest difference or the highest variation. So so we decided to go with these two components 1 and 2. And so as you can see in this slide, so here we did a heat map, the various heat maps for PC 1 to 12, and you can see, okay, this one is not, it's not that clear, but if you when we did this and you look at it maybe in pairwise comparing 1 and 2, 3 and 4, you can see that as the PC number increases, the number of genes like that are expressed. So the differential gene expression decreases as the PC number increases. So this shows that PC 1 and 2 are able to represent like a higher number of variable genes or differentially expressed genes. And then with the last one, which was about the clustering, where we use the UMAP in the clustering from what was in the R script. And then, so from here, you can see that the various, so we did the clustering based on the disease status, which was control and COVID-19 participants. And you can see that most of the cells are overlapping. That's for the control and then the COVID cases. So this shows that so maybe this can signify that it's actually the same cells, that if someone has COVID, it's actually the same cells which like in the healthy cells are also in the COVID patient, just as in the COVID patients, like there's the COVID virus has infected the cells, but also you can also see some variations as well with regard to some of the size of the clusters and the color. So yeah, it's not that clear, but if maybe to zoom it, you see that the some of the, so you see that at certain points there are two different colors. So this signifies maybe no variation. But if you see like places where you see only maybe the blue or the pink, that's when you see that those cells, those particular cells vary with regards to the control and then the COVID patients. Yes, so that's what it did.
Tina, thank you. That's very excellent. You learned a lot in a day. Do people have questions for Tina about what she just presented?
Yes, I have a question about this graph. So how can you find the number of components that account for approximately 75% and 85% using this graph? I don't know how to interpret like this graph.
Okay, so this
Yeah.
graph, Tina, do you want to try to answer it first?
No, please, you can go ahead.
Okay, I didn't want to talk over you, sorry. Okay, so this graph, the x-axis are the principal components, and the y-axis is this standard deviation, which is derived from the principal components analysis. For those of you who do like linear algebra type things in your math classes, it relates to like the eigenvalues of the factorization. But regardless, what it qualitatively means is the standard deviation there represents how much variance you've explained in your data. And if you remember how we do the principal component analysis is we try to find groups of genes that explain the most variance in the data, and that's our first principal component. And then we look orthogonally to that once we've kind of explained that variance, and that's our second principal component that will explain the remaining variance. And any remaining variance from there becomes our third principal component, and so forth. So each principal component is going to explain less and less of the variance in the data. So they're ranked by essentially how much variance they explain. So the first one, as Tina pointed out, explains a lot of the variance in the data, and the second one explains also a lot, but less. And you can see it monotonically decreases. So if you look at the area under this sort of under this curve, and think of it as adding up all the way to 1, then you can ask what fraction of the area is under the graph from principal component 1 to 2, or from 1 to 3, or from 1 to 4. And you see that most of the area under the curve is from principal components 1 to 10. So that means that like in that region, about, as Tina was saying, about 75% of the variance is explained. Or if you can think of it as 75% of the area of the curve is beneath principal components 1 to 10. But that area really represents like how much variance in your data you've explained. And you want to try to explain most of the variance in the data. Like let's say you were only explaining 10% of the variance in the data with the principal component you'd chosen. That means 90% of the variance just got kind of thrown out. And so you're going to lose a lot of the biological significance of your single cell data, versus if you are explaining like 90% or 95% of the variance, that's pretty good. That's not 100, but it's close to 100. And that the nice thing is that you're using far fewer principal components than you are genes. So remember, you started with 20,000 genes and you're moving to a smaller number of principal components. Like Tina was using 2 here, but you might want to use 10 or 15 as she's showing on the graph. So hopefully that explains it a little bit, but this is a common graph that's generated to help you choose the number of principal components.
Okay, yeah, I understand. Thank you.
Absolutely. Good question. Yeah. Any other questions for Tina? People want any clarifications on plots? Okay, this is great. Thanks so much, Tina. This is also like really well presented. Nice slides.
Thank you.
Great. Okay, so I think group 2 was the one that was experimenting with the different aspects of the clustering. Let me just check if that's true. Pretty sure that's what it was. Yeah. Is there anybody from group 2 here who's going to present? Who's able to present? And you can also, if you don't, for group 2, if you didn't make a presentation or anything, that's okay. If you can also talk through it. It doesn't need to be slides if you didn't have time, or if you just have questions about what we were even asking for. But yeah, is there anybody from group 2 here? Okay. Okay. We can. I'll put a message in the chat just in case people missed it, but I'll say if anyone from group 2 can present or talk through what they did, just let me know. Okay, because I can talk through it later, but I think I'd rather not do that. Okay, so is there anybody from group 3 who prepared slides or wants to talk through what they did? Okay, group 3, and how about group 4?
I just want to point out, we're all here to learn. So
Yeah.
there's, it doesn't have to be perfect. We're all here to learn from one another. So if there's somebody from group 2 or 3, raise your hand. Don't be shy. We're all very supportive.
Okay, how about we'll start with group, like none of these are really that chronological, so we can go right to group 4 and then we can circle back to group 2 and 3. So Akiko, that would be great if you could talk through, or thank you.
I think I'm gonna share code first. Okay, I'm quite new to R, but I'm gonna just go through some of the code. So for group 4, it's basically the results, the final stage, and then we looked at the gene expression in CD8 cells, or you can choose any cell types, between COVID-19 patients and then COVID negative or uninfected people. So first we looked at the number of cells for each cell type, and it's gonna be, so this one. Okay, so B cells, we have 4, and this is the table. And then we looked at the top 10 most expressed and genes in COVID-19 and then COVID negative patients. And so PCT is for COVID-19 individual, and PC2 is uninfected. So this is top 10 for COVID-19 people. And this is for top 10 genes expressed in CD8 cells, in COVID negative people. And And we finally expressed this one as a heat map. And so basically, so for this one, sorry, I also tested the squamous cells as well. I'm gonna show you
I'm
this.
trying to see if I can get your questions. I can finish my video. You can finish my session. I
Okay,
asked the
and
class
then
before. I sent
I
for the
think
class
someone
before.
is,
Oh, Chica, I think you might be mute. There we go. Okay, sorry about that. Akiko.
sorry, yeah.
Continue.
Yes. Okay, going back to this one. So we looked at COVID-19 and an infected patient for this one, squamous cells. And I also asked this question, what this one is. And then basically, because like you just saw this table, there are so many cells for ciliated cells, squamous cells. So for this one, instead of like screening all cells for squamous cells or ciliated cells, we just screened 200 cells and then generated heat map. And so this is basically the result for squamous cells. And then I also have the result for ciliated cells. So I'm gonna share my slide. Okay. So again, so this is the COVID-19 negative individual, and then this is COVID-19 infected individual. And then interestingly so this is for ciliated cells and then gene expressed in ciliated cells. And then there are some viral proteins that are expressed as well or included for analysis as well. And the first one is likely to be a reference sequence that I used. And then so the next one is nucleocapsid and then open reading frame 10. And as you can see, this viral protein is absent in control patient, but of course present in COVID-19 patient. And it looks like all open reading frame is related to antiviral innate immune response, such as interferon gamma and the interferon gamma induced gene or stimulated gene. And the bottom part is host proteins. And so there are so many genes as well. So basically for this group, it's for interferon gamma related gene and the HLA class 1 gene. Of course, these are upregulated in COVID-19 patient. And yeah, this is also like we discussed yesterday, this gene. So this is the X inactivated specific transcript for female X chromosome, because females have 2 chromosomes, and then so basically 1 chromosome is inactivated. So this gene like acts like that. So if you look at this row, so basically if it's red or expressed, it's basically these people are female, and then these people are male. And so if genes start with MT, this is basically mitochondrial genes, and they are expressed in both control and COVID. So it's really not like a characteristic. But in control patient, there are some genes that are specifically expressed or upregulated, and they are related to respiratory cilia function. So maybe like in COVID-19 patient, these cells are not really like functional. Yeah. And so this is the result for squamous cells as well. Some genes I actually don't know why like upregulated, but if you compare the expression between control and then COVID-19 patient, you can see that some genes are upregulated in control patient. And then when I looked at these genes, it looks like it's something to do with epithelial integrity. And then we also, it's very not distinct, but if you look at CCL5, more people are expressing CCL amongst COVID-19 patient. And then also this gene, also something to do with inflammatory response. Okay, that's it.
That's awesome.
Thank
Thanks,
you.
Akiko. Yeah, that's really great. I was impressed you kind of looked up all those the genes and how they're, some of them are cell type specific, like you found some things that are
Yeah.
specific to ciliated cells and some immune related responses, which makes sense in the COVID-19 infection, like the CCL type genes, like those are all different chemokine ligands. Very nice. Good job interpreting it. So just to summarize, what Akiko was doing in her group was comparing within cell subsets the effect of the COVID-19 or the SARS-CoV-2 infection and what transcriptional response there could be due to that. And so she found some viral genes that were high in the infected cells, immune-related genes, and also genes related to the cell type itself. So great job. Do people have any questions for Akiko? Okay.
I think I see a question actually, from Luis. Can you explain
Okay.
the expression of -2?
Yes. Yeah. Akiko, do you want to start with it, or do you have a sense?
Negative
Yeah.
From my understanding, yeah, I think it's down-regulated relative to housekeeping gene.
Yeah, it basically it is downregulated, or what I like to say is okay, so first I'll just make a point that I try to avoid saying the word upregulated or downregulated, even though that's often how we learn to talk about these things. I often like to say more like overexpressed or underexpressed, because the word regulation kind of implies some form of causation. Like we don't know that these genes are being regulated by whatever we're looking at. We just know that they're higher or lower. So it's a small point, but I think regulation is a stronger claim than increase or decrease in expression. But regardless of that, why are the values negative? So the reason that is because in the heat maps that we're showing, so Akiko, all those things Akiko was showing, those two plots, they're all called heat maps. What happens to each gene is, gene's expression can be from 0 to 1 to 2 to 3 to 4, and so forth. But what happens when we're displaying it in the heat map is we ran this function in Seurat called scale data. And what that does is it takes the gene whose expression runs from, say, 0 to 10, and it centers the expression of the gene around 0. So the cells that have more of the gene, or they're more highly expressing that gene, they'll have positive values, and cells that have lower expression of the gene, lower than the mean expression, they'll have negative values. So the negative there just means that in that cell or that group of cells, the gene is less expressed than within the average cell. But the real values of the expression are 0 to positive numbers. This is more just like a visualization thing. And why it's a good visualization thing is because now you have positive number, 0, and negative. So you can use like a 3-color scheme, and that's easier on the eyes. And it helps put everything kind of centered around 0, so that one of those colors can be for 0, one can be for positive numbers, and one can be for negative numbers. And if every gene is centered around the same value, it makes doing these color scales and just kind of comparing everything in one plot much easier. Versus if you don't do this centering of the data, you might have some genes that have very large values like 100, and some genes that have very small values like 2 or 3, and it's hard to kind of compare those on the same heat map because you have to assign a color to all those values. Okay. Any other questions for Akiko? That was a good question. Thank you, Louise. Okay. So is there anybody from groups 2 or 3 who would be interested in presenting what they found? Okay, so I think, how about I will really quickly go through it, and if anybody from group 2 or 3 wants to add on, they certainly can do so. Let's see. Okay, so let's start with group 3. Okay. Actually, that's a little more complicated. That one was just an easier one. Group 2. Okay, so group 2, there were 2 things that they were looking into. One is parameter choices, and one was comparing UMAPs and TS&Es. Okay, so let's just start with the parameter choices. So I'm just going to take the code and copy it into my console, one line at a time. So the first thing that the group had to do was try a range of k param from 2 to 100 and save your plot images. Let me remind you all what that k is. So we were building these nearest neighbors graphs to do the clustering. And so you first have to build this graph, and k is the number of nearest neighbors you look for. So k can be large or small. If you make k very small, that means you'll only look at your first nearest neighbor. And if you made k really large, like let's say you made it 20, then you would have connections to all of these cells, even though some of them are pretty far away. So there's this balance between very local, where I just make k1 and I only look at my very nearest neighbor. And if I make k20, things are very global and I'm looking at things that are pretty far away from me. So that will change the output of the clustering. So we can see that. So first, what we'll do is, let me show you the original UMAP. Actually, okay, yeah. So this is the original UMAP, to remind you all, that you kind of made yesterday. All the different cell types that have been annotated by the group that published this. Okay, and then what we're gonna do is we're gonna change k, and we're gonna make it really small. Like, let's make it 1 or 2. Okay, and then we'll do the clustering. And now we'll look at let me see. I don't think I have these. I don't know why,
The
but I don't
same
know why
thing
the
happened
plots
to
are.
me.
Yeah, I think it's that's why I can try saving it. Let's see. Sometimes if I just save it, I might fix it. Let's see. I don't know if it's the graphics device is just not cooperating.
Or maybe you can show them like on your or like on your
Oh god,
Oh,
that's
okay, there you
that's why
go.
it's so, this is
Okay,
why
okay.
it looks so crazy, because it's like let me see. Yeah, I can try that. Yeah, I don't know. I can try. Oh gosh. Okay, so you can see that there's what's happening here is that, let me see if I may, oh, it saved it. Let's see if I can open that save file. That's called UMAP subsets. Let's see. Okay, so this is kind of funny actually. Oh, wrong one. Just gonna reflexively, let me save it so it's, the width is way higher. It might not be, it might still not be wide enough. What's happening is that, okay, so we made the K really small. We made it 2. So you're really only looking in your local neighborhood. And what that leads to is a lot of clusters being made or identified. Yeah, you can see it's like it doesn't want to plot it. So let me, you know what, let's just make the K a little less ridiculous, just to see that, let's make it. This is why you do the experiment offline. Okay, let's see. So we made the K 4 this time. Okay, so now we can kind of look at this more easily. Okay. So you can see that there's like 27 clusters, and that's kind of a lot of clusters. You know, now let's compare it to when we did the default, which I think was 20. So there we have 27 clusters. And now when we increase the K to 20, we have 16 clusters. Anyone have a guess what will happen if I increase the K to like say like 50? Anyone have a guess? Or not even a guess, but a hunch? Or anybody from group 2 want to say?
Somebody said low range of clusters.
A lower number? Yeah. Less clusters. Yeah, exactly. Thanks, Mariama and Gertrude. Yep. That's exactly right. So now we have 13 clusters, and you know, we can't make the K more than the number of cells because you only have as many neighbors as there are cells in the data, but let's try it. We could do it 100. And so what I'm hoping you're seeing here is that like how you choose these parameters is going to affect your results. So now we have 10, 0 through 9, so 10 clusters when we make the K 100. So these are all things to think about when doing the analysis. And often you get the question like, what's the right number of clusters? Why does the cluster reduces the K? Okay, so let me show you this picture again. So if we make K very large, so now this circle is connected to all these other circles or cells. Now in the graph, all of these are connected. So they are more, so all in the graph, like all of these are going to be connected by edges. And what the clustering is trying to do is identify groups of cells that are connected by edges. So the more you have a group where there's just a lot of cells connected, the more likely those cells are going to be in a cluster. And so the larger, the more cells you have in a cluster, by definition, that means you're going to have fewer clusters. So when you make K large, it tends to result in clusters that have more cells in them. And because there's only a finite number of cells in your data, that means you're going to have fewer clusters to begin with. So if you had 1,000 cells and your average cluster size is 10, that means you'll have 100 clusters. Versus if your average cluster size is 100, then that means you'll have on average 10 clusters. So what happens with K is as you make it bigger, your cluster size just increases, tends to increase. Versus when you make K really small, like I'm only connected to my nearest neighbor, then the clusters become really small as well. And then that means on average, you're going to have many, many, many clusters. Good question. Does that, who asked that? Tina said, okay. Okay. At least one person got it. Anybody else have a question about that effect? Opening the wrong R. Okay. Really good. And then the other thing I wanted to show in this, and then if there's anybody from group this is group 2, who was able to run the UMAP and run the TS&E. If they want to explain it, happy to hear that, but I'll first run the UMAP. First, I'll go back to the normal clustering. Excuse me. Please, what is the essence or importance of the clusters? Okay, so good question, Chica. So the point of the clusters is, okay, so each of these clusters is probably going to represent some biologically informative cell subset in your data. So the overall goal of what we're doing here is to really identify cell types and cell states that are in these patients who have COVID-19 or these control people who are healthy or at least don't have COVID. So each grouping of cells is in a cluster, and those clusters consist of cells that are transcriptionally similar to one another. So they have similar gene expression profiles. And those gene expression profiles can be interpreted to understand what cell type these cells are coming from. So for instance, this cluster, that's number 13, like that looks very different from the rest. And cluster 4 kind of looks pretty distinct. And we really want to know what cell types these are. And to figure that out, what we do is we take cluster 13 and we compare it to all the other cells in all the other clusters at a gene by gene level. And we ask, is gene 1 in cluster 13 different from all these? Is gene 2 different? Is gene 3 different? And when we find the genes that are differentially expressed between clusters 13 and all the others, we get a hint about what that cell type could be because we have pre-existing knowledge about many of the cell types. So we can look at the actual, oh, I shouldn't have done that. Let me go back to, so we can look at the actual identities of the cells as determined by the authors of the study. So let me show you this side by side. So on the left are the clusters that you determine when doing the clustering, and these are just numbered because we don't know what they are. We don't know what they represent. Okay, you've seen this before. Here's cluster 13. And on the right are the cell type annotations determined by the authors. And what you can see is like, this cluster 13 is very different. This is a cell type, a special cell type called ionocytes. In cluster 4 here, in this sort of one of the green shades or sort of olive is squamous cells. And these green cells up here, these are T cells. And then this big group of cells here, they're annotated as ciliated cells, but you actually see that there's maybe subsets of ciliated cells, or at least you can cluster and identify something that looks like subsets. It takes a little more staring at the data to say if that's real or not. But what you can see is like the clusters are what we're using to determine essentially the cell types. So hopefully that gives you a little more concrete sense, Chica, about why the clusters are important and what we're trying to get out of them. But it's a good question. Okay, so okay, so we ran the, oh, there's another question. Thanks, it's clear. Great. Okay. So then the other thing we did in this was we ran a UMAP thing, and we can make okay, the UMAP's going to run. And so UMAP and TS&E are different ways to embed the data. So far, what we've shown in this little plot are UMAPs. That's the UMAP there. And then we can also show this as something called a TS&E, which is something that was very popular before the UMAP and became less popular once the UMAP came out, because the UMAP has some nice features about it, both visually and mathematically, that people like. So the TS&E, I think, takes a little longer to run. So we'll let it run in the background. Does anybody have a question about any of this so far? You know what, let me do the following. So here's the UMAP on the left and the TSNE on the right. And so what you can see is that the embeddings, how the cells are sort of ordered in this two-dimensional plane, they're pretty different. The UMAP, even kind of qualitatively, things look a little more distinct, a little more compact within these clusters. You know, the ciliated cells are taking up less space. The squamous cells, there's less just spreading around an area between similar cells. And depending on the parameter choices you make, you can get different looking plots as well for these UMAPs and TSNEs, and you can play around with that. It's just a matter of wrong one. It's just a matter of changing some of these things like perplexity or number of neighbors and so forth. And I think Sergio said this yesterday, but these are all calculated based, the UMAPs and the TSNEs, they're all calculated starting from your principal components analysis. Is there a difference between, so there is a difference in that they're both what's called nonlinear dimensional reduction techniques. And they make a lot of different assumptions about the data and what in the data they're trying to preserve as you go from high dimensions to low dimensions. So they're mathematically just very different. And then we did in the slides, we actually had a link about this. Let me find it for you. Right here. So there's a link you can follow in the slides that are on the GitHub called Understanding UMAP. I can put it in the chat. And that's very good because it gives you an interactive visualization of TSNE and UMAP, and you can play around with the parameters. Okay. And then, great. Are there any other questions? Is there anything to infer? Yeah, good question, Peter. So let me go back to. So the distance between the clusters. So like, is there a difference between the ionocytes and the ciliated cells versus ionocytes and T cells? And if you have to remember one answer to this, or the simplest answer is just no, there's no difference really between the distance between clusters. These techniques aren't very reliable when it comes to like globally how different cell types are, like very different cell types, like how more different is an ionocyte from a T cell, from a ciliated cell, from a squamous cell. Like these mathematical transformations don't really preserve those types of distances. What they're trying to preserve is local similarity for the most part. So things that are similar should appear close together in 2 dimensions, and things that are dissimilar should not appear next to one another. But how far apart they are is hard to know just from this type of embedding. Also, it's even if you could say what those distances are, it's not a super meaningful comparison, I think. Like usually we're not trying to ask, are B cells more related to T cells or to monocytes? Like you can answer that question, but it's not, it's more like there's not really a follow-up to that. It's more of an observational thing. There's nothing that you would never design a study really around answering that type of question. Really what you're trying to find are the cell types themselves, and then asking within cell types what's changing, or what are genes that define this novel cell type I found. But comparing like pairs of cell types and asking which are more or less related is sometimes that's a question, but it's usually not like I'm comparing B cells and epithelial cells. Like nobody would ask that. Okay, thanks, Peter. Any other questions? Okay. I think I want to be aware of the time because Christine, I think we have, I forget, we have till, I forget what it is in Greenwich Mean Time. Let me look.
Yeah, so we have summary and closing remarks, I guess right now.
Oh, right now. Okay, yeah, let's
But-
get to that.
We were
Um,
just gonna hold, and then after the summary and closing remarks, we were going to have office hours for anyone who has questions for the workshop leaders. It's kind of just more of a free form.
Okay.
So I don't want to rush you off, or if you still need to go through things.
I think we should just do the summary and closing remarks because and if people have questions, it doesn't have to be about whatever the other chunk is, group 3. We can ask those questions then, or yeah. So I think it would be better just to, because I think people also might need to, I don't know if anybody needs to hop off before in 15 minutes. So I don't know. Who's here? So Sergio is still here, I think.
And Sarah.
Sarah's here.
And I
Sarah.
Collins
Okay. Okay. Maybe
and Vincent.
yeah, Collins or Vincent, do you want to give any remarks? I mean, we can all say 1 or 2 things, but if there's any, maybe you have something you want to say.
Oh, thank you, or and thank you for taking them through this session. I think we've done a very good job. The training has been good. I think most people have learned a lot, and I'm grateful for all the trainers that have taken most parts of the sessions, the labs, and the talks. It was really great. We've learned a lot. Myself, I've learned a lot, and I'm sure the participants have also learned quite a lot. So we are really grateful to Christine and everyone on the planning team for everything they've done to ensure the success of this workshop. So thank you all.
Thanks a lot, Collins. It's very nice of you. Great. Sarah, is there anything you wanted to add on to that, or are you good? Okay, Sarah might be.
No,
Oh, there she
I'm
is.
here. Oh wait, so my screen is over there, so you might feel like I'm not looking at you. But yeah, thank you so much for all the people who have attended this workshop. I hope it was helpful, and I hope like it's a startup for you guys so you can start analyze your data. Thank you to all the leaders and Elliot and Christine. You guys did a really good job. I myself felt like I was learning a lot, also from other training leaders. And yeah, thank you so much.
Great, thanks, Sarah. Yeah, and likewise, I think thank you to Elliot and Christine for doing an amazing job organizing. This is like really hard, and Tracy too. I don't think she's on the call, but this is a really hard thing to organize. And thanks to all the people who did all the work to put together slides and labs, all the trainers. This is really amazing. And then I think what I'll just show is also, I posted this in the Slack, but I think it's useful. I posted in the Slack all these links, and I'll just show you them as a PDF, but what these are is kind of an organized set of things you can use going forward. So these are, the first 2 are just links to repositories where all the single cell tools are listed for computational analysis. Not all of them, but it's like a large number are organized here. So let's say you're interested in differential expression. you can look for tools that do differential expression. And so there's a nice like kind of organized way of finding things, cell classification and so forth. So those are some lists of tools. Then we have links to software packages. So in this course, this workshop, we use Seurat, but there's things in Python and so forth if you're interested. Collins, Vincent, and Dominic already sent you articles about the Human Cell Atlas, but there's some more linked here. A lot of people asked about further online resources for learning. So these are some of the best right here. There's 4 links here. These are just different courses. Like for instance, you can click on this one, OSCA, and I think it's, yeah. So this is like an online book, and it's organized. There's book contents, introduction. Yeah, so there's, I don't know why the links aren't, let's see. Yeah, here we go. Okay, so now we can see everything. Yeah, so now we can see everything. So there's feature selection, which we did yesterday, dimensionality reduction, clustering, marker gene detection, cell type annotation. And then if you get like, you know, more advanced stuff, there's different things about normalization, again, more information about clustering. And so there's, you know, lots of stuff to click around here and explore. And hopefully that will be informative. So there's these online courses. And then finally, where do you get data? So it takes a lot to, you know, a lot of resources to generate these data sets, but fortunately, and I think as you saw yesterday through the CZI presentation, there's a lot of data sets that are just publicly shared and organized. So for instance, we can go to this one, Tabula Sapiens. And this is hosted by the CZI. So you can launch, and this will be like, I think, 500,000 cells that will get loaded at some point. So there's a lot of just, yeah, it's 480,000. Okay, yeah. So, and we can color it by, let's say we'll color it by tissue type. So there we go. Okay, so there's lots of that in those links I just sent you. And I like this other one here from Sweden. This is interesting. It's sort of like a crowdsourced listing of single cell data sets. So this is, now we're in, this is the paper, excuse me, this is the paper. Well, I guess I can't, but this is basically a list of single cell papers that are out there. And then like information about the data sets, when they were collected, tissue, technique, and so forth, like just lots and lots of metadata. So you can find like, essentially, let's say you were interested in, you know, all the brain studies. So you just look for brain, you know, and these are all the tissue types. So there might be lots of data here that might be of interest to you. And there's, I mean, I don't know how many rows are here. You know, 1,700 rows, and people are still adding. Like the last one was from, I guess, September 23rd. Yeah, so that's also really useful, I found. Yeah, and let's see, anything else in those resources? Yeah, so that's gives you an overview of some of the resources that are there. Any are core, okay, so then, okay, I have a lot of things in the chat. Let's see. Any R course recommendations? Yeah, actually, I don't have this in here, but let me give you one. It's I think it's R Intro Data Science. R for data science, yeah. So this one actually I quite like. Because it's really based around exploring data and making plots. It's written by Hadley Wickham and this other person, but they did a lot of contributions to these very useful packages in R called TidyR. So if you can just go through this online book, and it's really focused around learning how to visualize data, but in the process you will learn like how R sort of works. And it's very well written. This is actually how I learned R many years ago. I just read this and then I figured it out from there, but this was a very good introduction, I thought. So I would start there. There's obviously things like Coursera, I'm sure, has introductions to R, and a lot of these things are free. Yeah, so let's see, what other question was there? Articles on gene regulation. Okay. So, thank you. Okay. Okay, so I have there's some. So there are some more science-y questions. So does anybody have any other general comments or things that they'd like covered? Otherwise, we're just going to switch it over to essentially, I think, an office hours type thing where we'll just answer some specific questions you still have. But I think if not, we'll kind of end the formal part of the course.
Thank you, Or. I just wanted to say thank you to all of the training leaders. They were very thoughtful and put a lot of time, energy, and effort into designing the course. This was, you were kind of our pilot group. So we will be sending an email out shortly with a link to a survey for feedback. We would really, really appreciate you taking the time to fill that survey out. It should take 5-10 minutes, but it's important for us to hear from you, especially since we'll be taking this on the road to Africa next year. So we want to know what you want to see more of, what wasn't so helpful. And please be honest, Elliot and I and the training leaders will read through every piece of feedback and try to be respectful of it and try to work it into the design of the courses for 2023. So I think this is all of our workshops. So we want you to participate in helping it get designed. So thank you to all of you for showing up and for participating. And we really hope to see a lot of you next year in person. As I mentioned, on day one, we will have an in-person workshop at the University of Ghana, February 20th through the 23rd. So more information will come on that soon. And if you are interested in participating in that, keep in touch with Elliot and me, as well as the other organizers. And then we're also looking at another potential site in Nigeria, I believe in May. Elliot, is that right?
I think June, but
Oh,
yeah.
sorry. So
Early summer.
So June 2023. And then we're also hoping that we can identify a site in maybe South Africa and North Africa, so we can make sure that, and East Africa as well, so we can get as many of you in person as possible for those workshops. Elliot, is there anything else that you want to say housekeeping wise?
No, just wanted to thank you all for joining us and participating. It's been really great. And, hope to meet some of you in person throughout, the next few years. But if not, also maybe in part of more of these virtual events. Thanks for joining.
Excellent. So, or I guess over to you and the team for office hours.
Yeah. Okay. So, yeah, thanks, everybody. So, and I'll just say it was all nice to meet all of you over Zoom, and really a pleasure to teach and hear about how you're doing with learning all these things. It's very exciting, and best wishes as you figure all these things out. It takes a lot of perseverance, but it's very doable. Great. So I think with that, you know, if you want to stick around, feel free to, but if you need to go, you don't have to stay. But I think there was just some questions in the chat about one, links to articles on gene regulation. So I might have something like that. Let me just look real fast. So there are ways to sort of infer gene regulatory networks from single cell data. We didn't really cover that in the course, but I can post. I'm just gonna post a review article for you in the Slack. Oops. And let's just do it in the resources. Okay, I just review on gene regulatory network inference. Sent. Okay. Okay, so I just posted an article that kind of talks about that a little bit. Okay, and then the other question was, oh, so Kiko, okay, we got that. It's in the Slack. And then there was another question on SiteSeek, I think. I don't know. Sarah, have you done SiteSeek at all? I can answer it, but I can. You've done it.
No, but I
Okay.
think Daniela has done
Oh.
a lot of them.
Oh, I don't think I saw Daniela this morning. Let me see. She might be doing lab work or yeah, I don't see her. Okay, so when using a tool like SiteSeek, is it useful to stain cells with flow? Yeah, so SiteSeek, Luis, what it does is you're saying there is you have antibodies against surface proteins, typically. And those surface proteins are often markers of cell type, or of some interesting phenotype you care about, but often people are using antibodies against like different immune cells. So CD19 or CD3 or CD45. And that does actually help you identify the cell types quite a bit, especially because some immune cells are a little hard to separate out at the transcriptional level. So like CD4 and CD8 cells, T cells sometimes look a bit similar transcriptionally. And so an antibody against CD4 and CD8, if it's quantified at the single cell level, can really help. It's not like the default way that people do RNA-seq, like we don't by default run them with the proteins and do site-seq, but if you have a specific kind of question where knowing protein expression levels of certain things would be helpful, then that's when you do site-seq typically. It hasn't been widely adopted, I would say, yeah, but it is interesting in that it can help with identifying cell types.
And if I can add something, I think regarding the question, if you can use also staining for flow before SiteSeq, and the only recommendation, it is possible, the only recommendation is try not to use the same clones from the antibodies from the SiteSeq than from the flow. Otherwise you will have competition and it wouldn't work.
Right, that's a great point, yeah. Yeah, thanks, Sergio. I didn't think of that. Let's see, thanks all speakers, thanks all. Let's see. This is good. Thank you very much. Thank you very much. Please, for how long do we have back? I don't know. Is Vincent still on the line? Yeah, Vincent, I think there's a question about how long you have access to the RStudio server for.
Yeah, I think he responded.
Oh, he did?
He said that.
Oh, for 2 more
it's
weeks.
gonna be, yeah.
Okay, thank you. Yeah, got it. Okay, great. And then the other thing is, you know, you can install R in RStudio as Sarah had shown, and then have everything locally running. Things will run both in Windows and in a Mac and in Linux for R, so it should work for all of you. You know, you can post messages to the Slack if you are having issues, and we can try to maybe troubleshoot a bit. If you do post an issue to Slack, just be as detailed as possible in terms of what's your operating system, the order of steps you did, and a screenshot of whatever error you're getting. So we don't have to ask you like 100 questions. So the more detailed as possible, if you have a problem installing anything, then we can maybe help you. It's like not a promise, because sometimes these things are really hard to figure out over virtual things, but also you might have colleagues who have run R before and can also maybe help you, who are closer in physical space. Yeah. Any other questions that people have? Or all the resources are in the Slack. Yeah. Okay, so I think let's wait another minute or two, but if there's nobody has questions in the next 2 minutes, we can take it to the Slack for the future things. And yeah. And hopefully you know, I really recommend that you try to maybe give yourself a break for a day or two from all this learning stuff, but maybe early next week, try to go back into the RStudio server and just rerun the scripts and see, you know, what you've learned and where the gaps are. Can you remind me at what point does the data go into cell by gene? So cell by gene, Luis, you can put essentially gene expression data that has a UMAP or a TSNE embedding or some sort of embedding. And that's all you need. I mean, ideally you would want some sort of annotation of the cell types, but strictly speaking, you don't need that. But and what's nice about cell by gene is you can look at expression of individual genes or groups of genes. And you can color things by the different metadata. So the more metadata you put in, like patient age or sex or disease status or, you know, course of treatment, like the more informative it tends to be. So it kind of, it's close to what we have at the end of this last lab that you did yesterday. Like once you've clustered the data and cleaned it up and, you know, done some annotations, that's usually when you put it into something like cell by gene. Like it's not the first step you would do in any kind of process. Yeah, good question. Yeah, and hopefully Gertrude, you're able to open up the things on Slack. I'm sorry, on GitHub. If you can't open them up, we can post PDFs of the lectures in the Slack channel too. Just let us know. That's easy. Any other questions that people have? Okay, great. So I think we'll then close out and hope
Great.
you guys all have a good you know, time learning, getting, you know, I hope you're keep learning this and get better and better at it.
Thank you, Or, and thank you to everyone who participated and helped to lead this workshop. Please stay in touch with us. Please use the Slack channels or feel free to email us directly. And with that said, I will now close the meeting and wish you the best of luck with your continued learning on these techniques. So thank you and stay in touch. Bye, everyone.
Bye.
Bye. Thank you.
Bye.